I am trying a new lecture today, and this lecture attempts to put together a whole lot of different themes in quality control. Quality control is a topic that has not gotten a lot of attention, and you kind of get the impression that a lot of people in the field of proteomics are vaguely embarrassed about it. So today we're going to try to talk about some of the issues, why that's come about, and what is the real state of affairs in this space. Now, as I mentioned, there's an article in your folders there from Bell et al., with the title showing on screen, that describes the current state of biological mass spectrometry in South Africa. I hope that I got the details of your sites correct. So if you see things that need to be improved about that, by all means, let me know. We'll try to get that correct in our roster of who is where in South Africa. I believe the current count for this paper was 18 different institutions that have mass spectrometry labs with available core services. Would you have believed that? 18 different labs in South Africa. So that is a substantially growing field in this area. The paper that this updated from 2008 had a substantially smaller number of sites. I think CSIR was just barely off the ground, and CPGR was kind of coming into fruition. But we've grown a lot. Quality control represents one of the ways that we establish our staying power. Now, one of the papers that I made accessible to you is a review article that I wrote for Clinical Biochemistry a few years back. I think that you'll find this article useful whether you work on the bench or you're working with the data after the fact. And one of the things that we detail there is that there are many ways in which variability can enter the results that we see from biological mass spec. So as we move ahead, I want you to think about where does quality control sit today? And in many respects, the first tutorial is going to reflect that theme. And then we can think about the dream. How do we get to a place where quality control systems provide us the information we need to make decisions? So the way that I'm going to organize this is to start with the question of reproducibility. A lot of people have been worried about this reproducibility crisis thing, and we are going to take that on head on. And I'm also going to talk more specifically about different papers that were produced by the organization CPTAC, the Clinical Proteomic Technology Assessment for Cancer, and its follower, the CPTAC-2, which was actually kind of a data production program. And so we'll talk about some of the papers that came from that to evaluate whether proteomics is in fact reproducible. From there, we're going to talk a little bit about putting a number to variability. So if you have a new experiment and you want to know, is this instrument conforming to what we call good performance, or is it really an outlier? Is it giving us far fewer tandem mass spectra, for example? Okay, so we'll talk about metrics that are appropriate for shotgun workflows. This is for data-dependent acquisition, if you're a thermo guy, or information-dependent acquisition if you're working with SciAx. Each of the vendors has a different name for this technology, but essentially sampling tandem mass spectra from the intense ions seen in mass spectrometry. I think by this point, everyone is pretty clued in on what data-dependent acquisition is, so I want to make sure everyone can think about it in those terms. So how can we generate metrics from an LC-MS experiment that can tell us whether the instrument is conforming with how it should behave? Okay, from there, what about the other folks? Right? Not everyone uses liquid chromatography, right? So a lot of folks are making use of MALDI still, and certainly it's a very viable workflow for getting quick turnaround on results. So are there systems that can help us do quality control in this space? And of course, we'll talk about some of the emerging flows as well, things like SWATH. How can you tell if a SWATH experiment is behaving itself? And from there, we're going to talk a little bit about the dream. And I'm going to talk about this in the context of several people that we want to help with quality control workflows. So we'll walk through that as we get to it. All right. The first thing that we should address is whether, in fact, we are in a reproducibility crisis in the field of proteomics. Would anyone like to vote on this one? Let's try a vote. I would like to see a show of hands for people who say proteomics is in a reproducibility crisis so far that nobody can trust our results. Oh dear. Okay, so I see a lot of people with comma PhD behind their names who say yes. Okay. Are we Are we overblowing it? Is it hype? Are proteomics results just as reliable as what you could get from transcriptomics? And we're not the only people who ever struggle with reproducibility. If you think that it's all kind of overblown, raise your hand. All right, so I see there's a perfect superset. The people who voted for the second option also voted for the first one. So it could be a crisis of confidence. Imagine if you're not a proteomicist. Imagine if you are Jolene biologist over in the Department of Entomology, insects, right? So imagine that you've shown up at the proteomics lab saying, I hear you guys can identify proteins. Can you help me with these crickets? Okay, so the person like that who has no exposure to LC, no exposure to MS, no exposure to tandem MS, or exposure to the statistics that are required to detect differences, does Jolene know everything she needs to know to feel comfortable that the result that comes from the proteomics lab will be trustworthy? Frequently, people who are coming to proteomics from the outside perceive us as a field that gets a different result every time they sniff a sample. So that is a worrisome thing, especially in a country where biologists are not accustomed to relying on the mass spec facility. They need to have education about what the likely source, what the likely degree of variability will be in the result, what kind of sensitivity is really feasible, because heaven help them, they may be reading papers from major laboratories that state you can identify every protein in a species in 30 minutes. This and other filthy lies have tainted us. So I think that it's very important that we educate potential users about what are the strengths and what are the weaknesses of our methods. So I appreciated this article by Finelli that was published this year. And you can see that we have gray as other or not classifiable publications. We have those in red that say, these are papers that talk about what the reproducibility crisis looks like. And then we have the blue, which are papers saying, you know, this reproducibility crisis thing is actually overblown. So do people get a lot of exposure to the idea that we're in a reproducibility crisis? Yes, they do. Must we, as the proteomic experts, have some sort of answer for people who come to us saying, well, I hear I can't trust a word you say, right? We need to be able to speak to that degree of concern. So CPTAC came about because National Cancer Institute was unsure that they should continue dumping money into proteomics. Isn't that scary? National Cancer Institute funds a lot of proteomics research. They decided that they would team up with the National Institute of Standards and Technology and a bunch of labs that decided to become part of this U24 program to test the stability, the reliability, the variability of proteomics. And they hired a bunch of labs to do this. The U24 funding was a very big part of what I did as a researcher at Vanderbilt. I spent 10 years being part of this program. So I wanted to highlight a few papers from this, and I hope you don't mind that 2 of them come from me, because I really have been focusing on the questions of repeatability and variability and reproducibility over time. So let's start with this simple question. Do we see the same peptides each time we shoot a yeast sample? Right? So we're just running LC MSMS on yeast lysate trypsin digest. Okay? So are there more proteins in a yeast digest than what you can see in a single LC MSMS? Is it a simple mixture? Is it complex? How many proteins are possible in a yeast? Saccharomyces cerevisiae. Size of the proteome for yeast is what? Do I hear 4,000? That's E. coli. Okay, E. coli is like 4,400, I think. But how about Saccharomyces cerevisiae, red yeast? Baker's yeast. What's that? Okay, we've got, I'm going to bracket you. Someone back there says 4,000, he says 8,000. Okay, so I said it was higher than 4,000. 8,000, do people want to go higher or lower on 8,000? You want to go higher than 8,000 proteins on yeast? Okay, does anyone want to go lower? Catherine says lower. I see a lower here. I'm going to go lower. I'm going to go lower. It's not 8,000. There are fewer than 8,000 proteins encoded by the yeast genome. 5,300. I'm going to go higher. It's about 6,200 if I remember. Is my number mostly accurate? Okay, if you just download the complete yeast proteome from Uniprot, you get about 6,000 proteins. Now, are all 6,000 proteins expressed all the time in yeast? No. Some are specific to the budding process. Some are specific to when they can't get all the glucose they want. There are all kinds of different stimuli to which a yeast responds, and gene expression, and thus which proteins are present, changes. How many, at the time of CPTAC, I'll just let you know that a single shot on say, an Orbivalos, which was a pretty hot new machine at the time, would see about 6,000 proteins. 600 proteins. 600 proteins. So would you argue that 600 proteins is a complete roster of all proteins currently expressed, or would you suggest it as a subset? Subset, okay, great. I'm glad we've got that kind of set of expectations in place. That'll help make more sense of this. So what we've done in this case is look at 3 different LTQs and 3 different Orbis. In this case, I think these were Orbi XL at the time. I may be mistaken on that. And what we've asked is how much does our list of distinct peptide sequences grow as we shoot the sample a second time, or a third time, or a fourth time, up to 6 times on the same instruments. And what we see is kind of curious, actually. We see that on the LTQ, the number of peptides we've gained after 6 shots is 140% larger than when we just shoot it once. Okay? We see that this curve is rising, but is the curve going exponential? Going exponential? It is not. It's not. Instead, we see it's plateauing. Right? Does everyone see the distinction? This means the number is rising with each added replicate the gains are rising. This means that the gains are diminishing as we add new replicates. So what we see is that LC-MS/MS is a sampling process. It is taking a whiff of the peptides that are available, some subset of which will get subjected to LC-MS/MS, will get subjected to MS/MS. All right, does that part make sense? There are more peptides available than what the instrument can see in any one shot. Okay. I hope this isn't just sort of dragging this topic through the mud. It's an important concept for everybody to get, I think. Okay, so what we see is that the LTQ gains peptides from additional replicates faster than does the Orbi. Isn't that weird? And a lot of this comes from the fact that the Orbitrap is only selecting for fragmentation those ions that meet particular charge state requirements. The LTQ will take a shot at anything that produces intensity, but that's not true of an Orbi. Most Orbis are configured to only collect tandem mass spectra from things with a defined isotope packet that is +2 or higher. This is how most people configure them. Similar tools exist in the QTOF world. So what we see is that because the Orbi is constraining itself to a smaller subset of these ions for MS/MS, its growth is actually slower than what you see on the LTQ. Okay, so this I think is a concept that a lot of people know, but I'm glad that we could talk about it because I think it's important to explain why. All right, now at the right, we talk about spectral counting. Do you know a spectral counter? Bridget knows a spectral counter. I'm a noisy spectral counter. I was part of the Vanderbilt team for years, and we almost always used spectral counting to decide if the proteins in cohort A were different than the proteins in cohort B. And this technique that we use is pretty straightforward, really. We simply ask how many spectra in each experiment matched to this protein. Pretty straightforward, right? So if we look at this diagram here, we see that on LTQ at site 73, we found a typical amount of variability occurred because in the first replicate, 28 spectra matched to this protein. In the second, 27. In the third, 29. In the fourth, 23. In the fifth, 25. In the sixth, 18. That is a normal degree of run-to-run variability for an LTQ for a yeast lysine. So is that good? I'm going to ask a tricky question. We see that replicate 4 had 23 spectra, that was the second lowest, and replicate 16 had 18 spectra. Are the peptides seen in replicate 4 a superset of those in replicate 6? What's that?
You hope so.
You hope so. But would somebody say it's not? I see someone saying it's not. Which spectra we see for a given protein will also change. So generally speaking, which peptides appear for a given protein from replicate to replicate will also change. You have no guarantee that these 23 spectra seen in replicate 4 are a superset of the spectra seen in replicate 6. It's quite unlikely, in fact. I suspect that the Venn diagram between them is somewhere around 50% shared peptides, which is a bit shocking. But spectral counting is not requiring that the same peptides be seen every time. It's saying that they're being sampled from the same kind of population. So even though these numbers might look reasonably stable, I mean, a range from 18 to 29 is a little worrisome, right? But this is normal variability. And something that should show up, because you've all had some sort of statistical training now. I saw you listening to Catherine. I saw that. So one of the things you probably realize is that your ability to detect whether or not a protein is truly different between two cohorts depends upon low variability within cohorts. Everyone got that? Your ability to detect a difference diminishes if you have high variability within cohorts. That's saying it the opposite way around, but I think the point is the same. So this is the kind of variability we can expect. Our statistical models had better be able to handle that, or we're doomed. All right. Now, I've started the conversation about cohorts and differentiation, and certainly people care a lot about that. In the repeatability study, we were looking at the same sample shot again and again and again on a bunch of different instruments, and that was fine. But in this case, we're going to start talking about the differentiation of cohorts. In this case, CPTAC 2 was in charge. Now, the instruments had gotten better. The fractionation had become much more significant. In the repeatability paper, we didn't use any fractionation except the LC in front of the MSMS. In CPTAC 2, though, we used a technique called basic reverse phase liquid chromatography, or BRPLC. They really, really needed a better name than that, but that's what we got. So, has anyone heard of basic reverse phase liquid chromatography? Oh. This is actually something that surprised me the most about South African mass spectrometry. Because the cost, the relative cost of running an LC-MS experiment is higher here as a fraction of your grant budget, we find that very few people are making use of techniques for fractionating before it goes to the mass spec. I see some number of people doing things like gel CMS. Do people know what gel CMS is? Not everyone. Okay. In gel CMS, you separate your proteins on a 1D gel. Now, instead of cutting out bands, we're going to cut it into regions, right? So maybe I have one long lane, and I'm going to cut that into 10 slices, irrespective of where the bands appear. And then each of those, we're going to cut into, I'm going to use the parlance of the paper, we will cut into 1 millimeter cubes. Does everyone know that? Does everyone recognize that for the lie it is? Is someone measuring how far apart the razor cuts are? No! You've got someone like Khadija sitting in the lab going, I hate this. That's what we do. And then those little bits of gel have an in-gel digest to release the peptides. Those then get separated. So that from one 1D gel, you now have 10 raw files. I'm using thermoterminology. Please forgive me that. I've been in thermos for too long. Okay, so in this case, we were using basic reverse phase liquid chromatography, which separates peptides in a reverse phase column at high pH, at basic pH. It separates it into many different fractions. We then pool the fractions in a special pattern, and that gives us a smaller number of samples that will then be subjected to acidic pH, RPLC. Okay, so that technique was being used, and we were reanalyzing again and again and again and again a whole bunch of mouse xenograft data. Does everyone know what a xenograft is? One of the creepier things that we do to mice in our laboratories is give them cancer. And do we give them nice little healthy mouse cancers? We do not. We give them human cancers. We take cells taken from tumors in human beings, inject them, and see that these mice then develop tumors made of human tissue inside their bodies. Okay, personally I find that awful, but it's what we do. I left that to the collaborators at St. Louis. What can I do? So, what we then had were 2 different human breast cancers that were being grown in 2 different kinds of mice. And because these particular tumors were really productive, we had an awful lot of protein representing these human tumors taken from mice. So, as a control, we use these 2 different kinds of tumors grown in mice in a comparative experiment run many, many times across many, many mass specs. In total, this study included 3 sites doing something like label-free analysis, and another 3 sites that were using iTRAQ labeled differentiation. So these are really different strategies. In iTRAQ, you use the reporter ion intensities to tell you whether this protein differs between this cohort and that. In spectral counting, you say, my goodness, the number of spectra we saw for this protein changed dramatically between the 2 cohorts. Okay, so what we're looking at then are lists of differences across many replicates. Don't worry, that door will shut. It sounds sort of like a backup alarm, you know, like I'm kind of in reverse trying to get into my space. He's got it. Thank you, sir. All right, so what each bar represents is a particular comparison of cohorts. So to use something like this is an Orbitrap Velos at site 65 using extracted ion chromatograms. This is the same instrument, this time using spectral counting. Okay, so that's kind of an odd kettle of fish. We have the same raw files, and we're examining them 2 different ways. We're doing it by spectral counting, but then we poke the same data through a system that can do extracted ion chromatograms. So what is the probably most widely used tool set in the world for label-free extracted ion chromatograms from shotgun data? I think I heard it. MaxQuant. Yes, the software you've heard me lambast. Well, I'm sorry. I just don't like it very much. It's just me. But it is, no doubt, very, very popular, and it has some really good training courses that go on all the time. So you can imagine then that the same data could be fed into spectral counting software or could be fed into extracted ion chromatograph software. And which proteins are going to seem differential will change depending on which avenue you use for evaluating it. So in this case, we see that for replicate A, we had about 130 genes that appeared to be differential on the basis of the proteomic data. And we see that this tiny little pip at the top is in red. If you look at the bottom, oh, it's in orange actually, sorry. We see that the orange ones are what I would call idiosyncratic. They're differences that appear in only that one experiment. But look how much the blue dwarfs the orange. Those blue indications are that genes that were found to be differential in this experiment were also found to be differential in at least one other. This is a very comforting figure. You might ask yourself, if I redo this experiment to differentiate this tissue from that, will I find the same proteins to be different? And the answer is, they won't be the same, but they will overlap a lot. There we see that we have experiments A through P. If I recall, I believe that was 16 different comparisons of xenograft 1 to xenograft 2. And we found that in the great majority of cases, if we found a gene to be differential in one experiment, we were also going to find it to be differential in another. That is a comforting message. So, before we just accede to everybody's claim that proteomics is irreproducible, I would point out that if you're doing your bioinformatics and biostatistics correctly, you're going to be quite reproducible in the differences that you find. All right, now up at the top, I was actually a little ruder when it came to these iTRAQ samples. You see, I guess it's quite small on this screen, but you can see the slide on your own computer. You have these slides in PDF format. You can see that I have samples A through E, and each of those has multiple comparisons within it. That's because in these samples we have 2 different replicates of one xenograft and 2 replicates of the other xenograft. So we can make 4 comparisons within in order to find those differences. But in the case of iTRAQ, I was much harsher. I said because the results between the results that take place within one 4-plex experiment are more reproducible, I'm only going to call these differences confirmed if they're found in a different 4-plex altogether. So that was pretty harsh. But look, iTRAQ still does pretty well in that context. So yes, when we do our statistics correctly, whether we're spectral counters or max quanters or iTRAQers, our differences certainly can be reproducible from one replicate to another. All right. Now, selected reaction monitoring is fascinating, and I think that we had a really interesting talk about the realities of what's accomplishable there yesterday. Thank you for that. But when we look at selected reaction monitoring in a critical way, we can ask if we send the same samples to many, many different laboratories, do we get quantification data that reproduce from site to site? Now imagine that somebody is running a 4000 QTRAP over here, and someone over here is running a TSQ Quantum, and someone else is running an Agilent. You're running an Agilent? We're making you run an Agilent today. You want your waters? Okay, she can run her waters, that's fine. If I send them all the same sample, do I have any reasonable expectation that their measurements of how much of this set of proteins is present in a mixture, that those amounts will be comparable in any way, shape, or form? We push this very hard. You can see that triple quads from different vendors at 8 different laboratories were each asked to analyze the same samples 4 times. We can see a whole series of different measurements here. So we have different studies that were run within these sites. We have different concentrations that were run. Along here, we're basically producing a calibration curve, right? We have increasing amounts of sample, and this reflects increasing amounts of measurement in response to that increase in actual concentration. So what we found is that the different sites, which represent all those different little colored stripes within those sections, are actually quite consistent. That's a very encouraging thing. Now we also talked about CVs. Did everyone catch the mention of CVs yesterday? CVs. Does everyone know what a CV is? Okay. Well, it's an important statistical concept, and probably the easiest way to describe it is to say it's the standard deviation divided by the mean. Does that bother you? I mean, I think there's a more subtle way to describe what a CV is, but that's kind of the sloppy way to do it. Okay. Does somebody want to give a more sophisticated explanation of that? What we are doing is evaluating how much variability there is for a given magnitude of result. So if you have a signal of 1,000, a variation of plus or minus 15 is a relatively small detail. But if you have a signal of 100 and you have a plus or minus 15, that's not so hot. Everyone sees that? So when we talk about CV values, we're asking how much do we get the numbers to bounce around? And if you recall from our discussion of spectral counts, we said that the larger the variation from replicate to replicate, the less our ability to find differences. So when we look at this in terms of CV, we see that even when we're down here at the very low end of concentration, we're able to stay, in most cases, below this 20% CV line. So this is the basis for a dictum that I repeat an awful lot, which is, if you're going to do a quantitative experiment, if you're going to want a quantitative output, you must do a quantitative experiment. Is shotgunning really, really quantitative? Is it really dialed in to limit variability as much as possible? Not even close. So it's frustrating to me to see a lot of people who want to be able to say, there's a 30% increase in this protein between these 2 cohorts, trying to do that by shotgunning. That's not a very good strategy. If you want to measure a ratio of 1.3 with very low, very small error bars on that, you should do a quantitative experiment. And we have an awful lot of triple quads in this country that are waiting for people to run samples on them, and a whole lot of orbitraps and Q exactives that have a 3-month backlog. You should probably think in starting your experimental design, am I seeking a quantitative result from it? If so, and I already know the suspects, the proteins that are most likely to be differential in these cohorts, you could just make your plan to be, build your plan around a triple quad. Okay, well, I will move ahead with that. So we're going to talk then about some methods for shotgun quality control. This section relates very strongly to what we're going to do in the before lunch session. I had a cold a couple weeks back and my throat is still not in fighting shape. We're going to start with a discussion of defined samples and manual evaluation, which is something that happens in core facilities around the world every day. And we'll actually go through that process together. That'll be a lot of fun. From there, we can talk about an early attempt to try to put numbers on the quality achieved by a shotgun experiment by Paul Rudnick and friends. Then we'll talk about some software that we're going to be using today. This is the QuaMeter IDFree mode. And we're also going to pause to think a little bit about iTRAQ. A friend of mine spends a lot of time thinking about iTRAQ-elation and how to evaluate it. And so we're going to talk about what he's built, what Karl Clauser has built into his Spectrum Mill software that's made available by Agilent. Okay. One of the most common strategies people use to evaluate whether their instrument is happy or not is to shoot a sample with defined content. Defined content. So one of the simplest things that you'll find people doing is to shoot a sample of digested BSA. Does everyone know what BSA is? Okay. Apparently, the world has quite a lot of cow blood, and cow blood's #1 protein is BSA. So it's really cheap to get your hands on a whole pile of this stuff. If you buy a few micrograms of this, digest it up, and aliquot it off, you can have a freezer full of this stuff and have the same digest to draw from for months at a time on your instrument. So we frequently find that people will shoot these defined mixtures of proteins. Maybe it's one protein. If someone's paying a lot of money for it, they might be willing to buy something like the Sigma Universal Proteomics Standard. It has 48 human proteins in equimolar mixture. So that's much more expensive. Now, some people will even step beyond defined mixtures. One of the quality control samples you can get is a total cell lysate, right? Maybe you shoot total E. coli proteomes, or total H. flu proteome, or total yeast proteome. This is all possible. But the kinds of analyses that are feasible when you have a defined content is quite different from those that you have when you have 1,000 proteins in the sample. So your QC strategy must reflect what kind of sample you're shooting. Okay, so remember that we discussed shotgun proteomics as a sampling process. So the instrument is going to produce a tandem mass spectrum of some subset of all the peptide ions that are available. So are the probabilities of detection for individual peptides the same? I see a no. Does anyone want to second the no? Is the probability of detecting each peptide in a mixture the same? Ah, there we go. I just need to say it differently. Okay, thank you for that. All right, so let us think about how BSA, we'll just stick with a very simple sample. Is every tryptic peptide produced from BSA equally likely to be detected in an instrument? No. Okay, so there are lots of reasons why they're going to differ. Trypsin, so imagine that BSA has some sort of complex 3D structure. I don't know what that looks like off the top of my head, but it's complex, right? So one of the first things we do is try to denature this protein and break apart all the disulfide bonds. We're trying to get this globular thing to stretch out so that we can start whacking at it. And if you find that some parts of the protein unfold more easily than others, the parts that unfold easily are going to be more accessible to trypsin. Is trypsin perfect? Trypsin is not perfect. And trypsin will, under certain circumstances, skip over a place that looks like a legit cutting site. Maybe you have too many arginines and lysines close together. Maybe you have, an aspartate right next to it. Maybe you have a proline right next to it. I mean, prolines, that's going to just abolish the digestion there. So it's quite likely that different parts of the protein will unfold differently, and different parts of the protein will cleave up differently as well. That means, just even from those two principles alone, we already have some peptides that are formed at much higher probability than others. Something with 4 missed cleavages in it, is that likely? Less so. Less so. So something that is a fully tryptic peptide is much more likely to be produced by the digest process than something that is erroneously cut someplace where there isn't an arginine or lysine at all. Next, we have the problem of ionization. We find that some peptide ions have these great big catcher's mitts, these arginines to grab the prolines and ionize beautifully, while others have very little basic residue content at all, and they don't ionize well. Right? So there are lots of reasons why some peptides for a protein are available at high probability and others that are available at low probability. And some of these are group phenomena. If you have a peptide that comes out at the sweet spot in the chromatography, it's trying to compete for charge and compete for attention from the DDA algorithm, all with a whole bunch of other peptides all clamoring for attention at the same time. If you have 100 peptides that are eluting in the same instant of time, the probability that any one of them will always get selected for tandem mass spectrometry is quite low. This gets even weirder when you start looking at mixtures of proteins, because now a protein that's present at a lower concentration may have a peptide that's found with very high probability that can outcompete a low probability peptide for a very major protein. So you can end up with all kinds of mix-ups along the way. Okay, so if the sensitivity of your instrument is reduced for whatever reason, you know, maybe the buffer is old, maybe you're getting an unsteady signal off of your ion source, I don't even know. Some of the peptides that you might hope to see in a good experiment will vanish. You won't see them at all because they're not able to, they're at too low a signal to noise to compete well to become tandem mass spectra. That's rough. Now, next up, your column may be compromised. One of the things that my friends at NIST frequently told me was that if there's a variability problem and the column or the electrospray or the buffers or the mass spec might be at fault, it's always the column. So they don't have very great perceptions of the robustness of the columns that we use in mass spec. That's neither here nor there. If your column is compromised, its ability to separate one peptide ion from another in retention time will probably be compromised. And that will lead to a diminishment in what you see. Finally, if your mass analyzer is out of calibration, the mass to charge of peptides might have a higher mass error. And sometimes, for some classes of instruments, this kind of loss of mass accuracy can occur in the course of the experiment, that you can see different mass accuracy values early than you do late in a single width file, for example. Okay, so there are lots of things that can possibly alter what we see in the peptides we observe. And we will probably think to ourselves, I know that this sample contains these peptides, and I should see them all the time. For each of them, we can have a mass value, or a mass to charge value, and we can have a retention time value, and we tend to pay attention to both of those. A lot of people tend to take a sort of relaxed attitude about proteomics, and when they do their QC, they focus on identifications. So why would you do that? You might do that because for a lot of people, the end product of shotgun tandem mass spectrometry is a protein and peptide list. So if that's what you really care about, you might be able to make some sort of assumption that all these things have to work well for us to get a good protein or peptide count from the standard we're shooting, from this defined sample. So in a case like that, you might try to just look at the IDs that result. Now, why is that challenging? We start with the fact that if you're trying to make an instant decision right after this defined mixture has been shot about whether your instrument is happy or sad, having to wait an hour for a database search is kind of lame. We would like not to have to wait for that. So database search is not the fastest software on the planet. It's a whole lot better than it was when I started in this field, but database search takes time. And if we want to make an instant decision when the defined sample has been shot, this is not such a great option for us. Next, protein counts and sequence coverages are relatively insensitive to variation. And I'm going to just make a really simple point about this. The unit of identification in a tandem mass spectrometry experiment is not the protein. Not unless you're a top-down lab. If you're Neil Kelleher, you can ignore what I just said. Neil, I mean it. So, I'm talking to the camera. Neil's in Chicago. Don't worry about it. He's doing fine. So, if you're doing top-down measurement of proteins, then those proteins are a fine thing to measure. But that's not what we do in shotgun proteomics. In shotgunning, we're looking at peptides. So if your QC is built around proteins, you're already several steps informatically away from the information you're actually producing. Okay. And finally, more PSMs won't ensure more peptides. Do you see what I did? I just used jargon there. Ashwell, I'm going to call you out on this one. I used the term PSM. What on earth did I mean? Okay, all right. John?
I've seen it in
this is what happens when informaticists start bleeding, start drinking their own Kool-Aid. I got to be careful about this.
Occurrences of that particular peptide signal in the sample.
Yes, that is right. Okay, so a peptide spectrum match. Right? A spectrum is just a spectrum. But if you assign an identity to it, you have created a peptide spectrum match, a PSM. Informaticists use this term all the time, and we assume everybody in the field does. That's really a dangerous assumption. So thank you guys for correcting me on that one. A PSM is a peptide spectrum match. Now, I've written something controversial up here. More PSMs will not ensure more peptides have been identified. Why could that be true?
So if he's just matching the same peptide number.
On the nose. On the nose. Okay, so in the tutorial, we're going to look at a couple examples of this, but it could be that despite the best efforts of data-dependent acquisition, we still capture many snapshots of exactly the same spectrum. It's the same peptide, it's the same charge state, it's the same set of PTMs, and we see 5 copies of it in a raw file. The software is trying not to do that. It's trying to avoid taking repeated snapshots of the same thing. Sometimes that's what it ends up doing anyway. So we're going to talk about that as we move ahead. So more PSMs do not ensure that you've seen more peptides. And I argue, and maybe I'm solo on this one, but I argue that peptides are the biological information we produce from these samples. Okay. Now, Rudnick et al. Thought that it would be really valuable if we could put numbers on a raw file that's been produced for some mixture. But along the way, he realized that there are many possible contributors to the variability we see in a given LCMS-MS experiment. So he tried to draw a little diagram here to help us with that. So over here, we have this little cartoon of an autosampler and pump. I think that this reflects the minimalist style very nicely. Many autosamplers have sort of a rotary thing going on in them, but yeah, the box with a box sitting on top of it is probably a pretty good abstraction. So I'm going to say to Paul, artistically, it's kind of a scratch, but from a schematic point of view, I like it. So we see that we have all of these different metrics concerning chromatography, because what comes after the autosampler? Okay, some Fritz and some nonsense, some tubing, and finally we get to an LC column, right? So we're going to lump together the behavior of the autosampler and pump and the LC column. We also, did I hear a comment? I thought I might, okay. When something comes out of the column, it hits the cattle prod, right? Do people use the term cattle prod here? Oh, okay. Sometimes I resort to Midwestern expressions. I hope you'll forgive me. Occasionally, you're trying to make sure that your cattle keep moving on the trail to market. And while you could just sort of swat them with your hat, some cows are a little resistant. So in the later era of the cowboy times, the cattle prod was invented, which adds a little voltage on an electrode that you could poke the cow with, which would cause it to move a little more aggressively. That's a cattle prod. So when I think of these peptides coming off the column, I think to myself of their nasty surprise as they get hit by 3,000 volts, right? Okay, so blam, electrospray, so the ion source. We're going to have some metrics that we can use to characterize those. Inside the mass spectrometer, magic happens. But it's a lot of different kinds of magic. So we're going to have things like MS1-based metrics, dynamic sampling-based metrics. Did you see what he did here? Dynamic sampling. Isn't that a nice name? So dynamic sampling is yet another name for data-dependent acquisition or information-dependent acquisition. Dynamic sampling. Which ions get sampled as tandem mass spectra? So we're going to characterize the MS scans. We're going to characterize which ions get selected for tandem mass spec. We're going to characterize the tandem mass spectra that result. And then, because these spectra get tossed off to a computer, we're going to also build in peptide identifications as a kind of information collected in these QC experiments. All right, and that was published back in 2010. And I have to say, it was kind of an interesting process. This is a CPTAC paper, and a lot of people don't actually think of it as that. But in fact, NIST had developed these metrics and we're using them routinely in-house for quite a long time. And it was only during CPTAC that we said, you know, these metrics really need to be made publicly available for everybody. So this paper came about, and it I think has, in a lot of respects, reflected the start of quality control metrics for shotguns. Very worth talking about. Okay. One of the things that I did while I was still at Vanderbilt was the creation of a tool called QuaMeter. QuaMeter started sort of under protest. I had a friend of mine, Lorenzo Fega, who had been working with the NIST MSQC software. This is the software stack you could get from NIST to generate these metrics from raw files. And he had been trying to make this thing work. After a while, he got everything installed properly, held his mouth just so, and he got tables of metrics out of raw files. But sometimes, something squirrely was going on. He would look at the metrics for files, and he would see something that made it look like a real outlier, but when he looked at the raw file that it represented, there was nothing wrong with it. So this was bothersome to us. If you're producing quality metrics, you don't want to create outlier metrics if in fact the file is just fine. So we thought, wouldn't it be useful if we were to try to recreate the set of metrics that Paul Rudnick's software does, except using our ProteoWizard codebase? One of the things that this would allow us to do is to produce these metrics from WIF files or from Agilent files or from Bruckers. Thought that would be useful. So we wrote the software called QuaMeter to do that. And you can see that there's a kind of a complex workflow here. We start with AB SciEx and Waters files being handled differently than those from Thermo, Brucker, and Agilent. So I have to say, it's been challenging over time to be able to use data from Waters instruments and from SciEx instruments as easily as we can the data from Thermo instruments. And part of this is because Thermo made a pretty hefty investment early on in getting the open source community to support their instruments. And some of the other companies were a little slower on that. So what we found is that we needed to be able to produce high quality peak lists from SciEx and Waters instruments. So my lab ended up creating a whole new bit of code called Can't Wait to peak list these things for us within the ProteoWizard library. Okay, so now that we can get all of these into an MZML format, we can provide that along with a FASTA file. Oh, FASTA file. FASTA file. A FASTA file is a database. Do you know what a FASTA file is? Yeah. We got a few people who do. Do you know what a FASTA file is? Okay, great. So a lot of people when they hear a database think, oh my gosh, this is something complex. Ultimately, FASTA files are just text files. Big text files, yes, and they're delimited text files, but they're just text. So we need to have some list of all proteins that are possible in this particular experiment. Great. So now we're going to run those through a search engine. Now I've written mirror match here, but I'm doing that because my team wrote it. Okay, this could be Sequest, this could be Andromeda if you're in for that kind of thing. And out the backside pops an MZ-identML. Some of you will have heard of that, some of you will not. Often we see people using PepXML as a way to represent the PSMs, the peptide spectrum matches, that result from this database search. Then we figure out which of these IDs we actually trust through ID Picker and feeding the filtered identification set and the original data to QuaMeter, we can kick out a set of metrics. So kind of a long pipeline, right? We already mentioned that the database search takes some time. All right. So I found myself thinking, why do we really have to do database search on these files to make sense of them? Is it possible to get any sort of useful information from these files if you don't identify them first? That was kind of a weird opinion, but I was looking for something really fast. I wanted to, my goal in my mind was 5 minutes. 5 minutes after the run is complete, can I tell whether things are looking square or not? So, in my mind, I wanted to throw away all of that business with the database search, throw away the database search filtering, and just process against the MZML object itself. So our QuaMeter IDFree software, the paper of which shows up in the files on your computers, is our ability to, gave the ability to examine things like chromatography, etc., without waiting on identifications. QuaMeter is pretty fast. Let me see. About how long does it take to run if you've got the file local? 5 minutes on a raw file?
Depends on your computer.
That's true. That's true. So if you're dealing with an i3, not so great. If you're dealing with files on a network server, definitely not so great. But if you've got the file locally and you've got something like an i7 to throw at it, you're going to have your result pretty quickly. So all of this was an attempt to get at a useful set of metrics right away. And you can see we published this in 2014. So what are these metrics and how could they possibly tell us anything about the data? I'm going to start with some generalities and then sort of talk about some specifics. The list of metrics is present in the second directory for the tutorial after lunch today. So what we see is that we're generating 40 different metrics, and similar to Paul Rudnick's strategy, we've categorized them into extracted ion chromatograms, retention times, the mass spectrometry-based metrics, and tandem mass spectrometry metrics. We felt that in many cases, any discussion of intensity, such as intensity ratios, really had to take place in the log scale. Because in a lot of cases, if you're looking for things like normal distribution, taking the log of intensity rather than taking intensity itself is going to give you a more stable result. We also spent a lot of emphasis on quartiles, and I'll show you some examples of that in a bit. So here's the first of our metrics, the XIC, which is shorthand for extracted ion chromatogram. FWHM, full width at half max. We'll come back to that concept in a bit. And Q2, any statisticians in the room? What's that? The second quartile, which is often called the median. Nice, nice. Okay, so we wanted the median peak width of extracted ion chromatograms. The name actually means something, isn't that handy? All right, so this is to give us the median of peak widths as a function of time, on retention times. Next up, I'm sorry, I'm standing in front of my own slides and making it hard for anyone else to see it, but we have the RT-TIC quartiles. So I'm going to have 4 different metrics that help me to evaluate the retention time, the capture of total ion current as a function of retention time. And that's going to be clearer on the next slide, so I'll come to that. MS1 density, Q2. Here we're looking at the median number of peaks appearing in MS1 scans. And finally, the MS2 frequency max. I think of this one as the burger counter, but I realize that's kind of an old thing to say. Back in the very old days, say the 1970s and 80s, McDonald's restaurants in the United States used to have a sign out front that would say how many billions of hamburgers had been served by the chain. 40, I think, now if you ever see such a sign, it just says billions and billions served. But in the old days, the numbers were a little smaller. So this burger counter is your bragging number. When you tell people how rapidly your instrument can collect tandem mass spectra, the MS2 frequency maximum will tell you the fastest rate of MSMS acquisition in the course of the, say, 90-minute experiment. Okay, so those are some examples of the kind of metrics. Now, I know that a lot of people have strong feelings about command line software. Does everyone hate command line software? Does everyone want a pretty user interface? We didn't write a pretty user interface. We really didn't. But to run the software, you say QuaMeter star.raw, meaning all the raw files in this directory, using the metrics type IDFree, and output the results to metrics.tsv. It's not bad. It's not bad as command lines go. We'll look at a much worse one in a while. Have you ever run a database search engine from the command line? You'll see what that looks like later on. Okay, so this is how you run the software. And now I want to try to explain some of these quartile metrics. Where do the peptides appear in your 90-minute experiment on your 90-minute shotgun experiment? Are they spread uniformly throughout the 90 minutes? Any yeses? Any no? I've got some people who have doubts about that claim. Over here, they're doubting that peptides are found uniformly throughout the 90-minute experiment. John, where do they show up for you?
Well, obviously, in a specific zone which tends to make mobile phase composition.
Okay.
So a lot of coming out around, let's say, 20 to 40%.
Okay, good. Right. The sweet spot.
Sweet spot, yeah.
We gradually ramp up the hydrophobicity at a slow rate through the course of the middle section of the raw file. And then we generally raise the hydrophobicity very much to kick everything else off the column. The peptides come off in this slow gradient, the shallow part. And some parts of it are very, very, see a very large diversity of peptides coming off all at once. So you might expect then that instead of finding tandem mass spectra, identified tandem mass spectra, just showing up at a uniform distribution across the experiment time, you see that they sort of bunch up in the middle, that your IDs usually show up really quickly in the middle. So one of the things that we can do is to represent the entire run as a timeline, and now we put in a slash everywhere we collected a tandem mass spectrum. So we know the total number of tandem mass spectra, maybe it's 10,000, let's say. So we can say what was the interval required for the first 2,500 tandem mass spectra events? What was the time interval for the second 2,500 tandem mass spectrometry events? How much time was required for the third? And how much time was required for the fourth? So this distribution helps you to understand the extent to which you had a lopsided generation of tandem mass spectra as a response to time. So, sorry? Thought I heard a question. I just coughed there. So what we see then is that quartile 2 and quartile 3 tend to be relatively short, and quartile 1 and quartile 4 tend to be rather long, reflecting the fact that that's not the sweet spot for where peptides show up. Okay, so you can characterize the chromatography, you can characterize the events that occur with respect to chromatography. So a totally uniform distribution would give you the values 25%, 25%, 25%, and 25%. In practice, we generally see that quartiles 2 and 3 are much shorter. Of course, it's all fine and good to make numbers from raw files, but those numbers may be actually useless to us. What if those numbers tell us very little about whether this file contains lots of identifiable spectra? So we needed to have some way to evaluate that. And I worked with a statistician at the University of Cincinnati named Xie Wang. And Xie is actually just amazing. She's done quite a lot to adapt what she already knows about biostatistics to this context of analytical chemistry. And one of the things that she found was that the metrics we were generating from QuaMeter have a lot of correlation built into them. And whenever you have a lot of correlation in the metrics that you're feeding to these decision systems, it's probably best to try to beat it out of them before you try to make decisions. Hopefully, principal components analysis gives us that kind of capability. So principal component analysis will be the highlight of the afternoon session in R. So we're all going to run a robust PCA in R this afternoon. So what are the things you can do with that? We start with dissimilarity assessment. That's kind of a silly name for it. What we mostly think about is the use of these metrics to find outliers. We want to find files that don't look like the others. Did you guys grow up with Sesame Street? There was always a little song associated with One of these things, the other. One of these things is not the same. You ever notice? Okay, there we go. It's a great song. You should look it up on YouTube. So these dissimilarity assessment helps us to spot those files that produce metrics unlike the other files that we've got in our analysis. So dissimilarity assessment. Batch effects. Do people know what batch effects are? A few people do. That's good. Batch effects happen because occasionally we have too many experiments to do all at once on our instrument. And it may be that you have to run the first half of the experiments this week and the other half of the experiments next week. Or maybe you're dealing with a really large-scale experiment in which your experimental acquisitions are scattered over months of time. The example we look at this afternoon is an experiment that spanned 8 months of continuous operation of an orbivalos, 1,425 raw files. So it's a pretty big data set we're going to be working with. So in a case like that, we want to know if the stuff we ran in February is at all comparable to the stuff that we ran in April. That's a pretty scary proposition for a lot of statisticians. So batch effect recognition comes into play for that. And finally, when you do find variability, what does it come from? What does it come from? And so for that, we have which sources of variability are contributing the most. So our paper gave examples of all 3 of these from the data sets we've provided. Okay. Now, Spectrum Mill is the software created by Karl Clauser for Agilent. Have you guys heard the name Karl Clauser before? Karl Clauser is actually a brilliant bioinformatician over at Broad Institute. He's had an awful lot to do with a lot of the growth of molecular and cellular proteomics. He was one of the people behind these publication guidelines that everyone loves so much when it comes time to publish their papers. But he created Spectrum Mill back when he was working in San Francisco, if I remember. So Spectrum Mill is a very weird search engine. It's got an awful lot of peculiarities about it. And I've listed just a few of these. Among these possibilities, the software can interpret tandem mass spectra partially in order to fuel its database search. It has a very chemically derived scoring system that reflects how peptides break. It can also do things like homology searching. What if you allow one amino acid in this peptide to be changed? Can you get a better match for it than you can if you leave that out? So it does quite a lot. One of the things that it does very well is deal with iTRAQ data. One of the things that Karl decided upon was that he wanted to be able to see raw data in order to be able to produce the best peak lists possible. You know, when you generate a tandem mass spectrum in, say, a thermo instrument, typically the precursor that gets written for it is the M/Z in the preceding MS scan. But you might realize, if you think about it, that you could look at the data over many successive MS scans and see slight variations in the MS each time and slight changes in the intensity each time and use that to improve your estimate of the peptide's mass to charge value. So pretty impressive, I think. So iTRAQ labeling has already been introduced, so I'm not really going to dive into that much more. But we haven't talked about why isobaric tags could lead to more variability in the data we collect. So I would point out that we have a model peptide here. Does everyone recognize NCC? Do we have any Star Trek fans? Star Trek fans? Oh good, we have a Star Trek fan? She's hiding, don't worry about it. Any Star Trek fans over here? No one likes Star Trek? This is very un-American of you. All right, that's fine. So, it was meant to be. So, Star Trek has Captain Kirk and friends flying around in a spaceship labeled the NCC 1701. So every time you watch Star Trek, you should take as a reminder, that's a peptide backbone. NCC. Right? Here it is. NCC. There we go. And then we've got this long thing standing out here. What on earth have I drawn here? That's an alpha carbon. And this is? It's a side chain. Yes, it is a side chain. Very good. Does anyone recognize it? Oh, I'm in the way. Lysine. Very nice. Who was that? Well done, sir. Right, so this is a lysine, and what it has in common with the N-terminus of the peptide is called a primary amine, right? This is a primary amine, this is a primary amine, and iTRAQ sticks to primary amines. So when you use the isobaric tagging strategy, you're not going to mark just lysines, you're also going to mark N-termini. Okay, however, different things that you do in applying the iTRAQ label can lead to variability in how it sticks to these peptides. So, one of the questions that you can ask is, for what fraction of the identified tandem mass spectra do you observe an iTRAQ reagent has correctly affixed itself to the N-terminus? To what extent have lysines appeared without having an isobaric reagent attached? Those represent errors, a contribution to variability. If you can identify a peptide and it has no iTRAQ reagent stuck to it whatsoever, you should be worried, right? So, being able to quantify that is quite valuable. We can evaluate the extent to which incorporation of the iTRAQ label is incomplete. Next. What happens if the reporter ion at 114 is always just a little bit taller than the reporter ion at 115? Does that mean your biology, that the samples that contribute the 114 labels are present at a higher concentration than the 115? We don't know. If you don't actually look at the distribution of the iTRAQ label intensities, your ability to know whether this one is high or low is diminished. So being able to characterize the ratio of individual reporter ions to the largest reporter ion in that spectrum gives us a way to characterize this over large populations of molecules. Okay. And finally, what was the median signal-to-noise ratio for reporter ion intensities? You might say, after I identify this peptide, I know that these are Y ions, these are B ions, these are potentially A ions, these are water losses, this is the precursor minus blah, 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 and these are the iTRAQ reporter intensities. Anything else, we're going to call noise. So then we can ask, are these reporter ions actually standing out of the noise, or are we just mistakenly calling something that happens to be an iTRAQ reporter ion, MOPZ, a reporter ion, when it isn't? Worrisome stuff. Are there any low mass ions that are produced when a peptide breaks apart? Okay, what's the smallest ion you can think of? Somebody want to say Y1? Okay, what's the mass of Y1? Y1 represents the last peptide bond breaking, and it's the C-terminus. And because these are tryptic peptides, that's either going to be a solo lysine or a solo arginine, right? So those show up at 147 and 175. So if you're dealing with a lot of tandem mass spectra without iTRAQ, you should see ions at 174, 147, or 175 and 147, I'm sorry. But are those the smallest fragments when we blow apart a peptide? I see peptide fragmentation as a favorite topic of this group. That's okay. I want you to think about something called an ammonium ion. Not ammonium, ammonium, sorry. These ammonium ions are representative of whether or not particular amino acids are present in this peptide. They don't tell you about its position within the peptide because they kind of represent like the side chain being blown off. So these ammonium ions show up a lot in HCD spectra, and some of them are awfully close to where these reporter ions show up. So if you're dealing with iTRAQ quantitation, it's useful for you to be aware of all the ways that this experiment could go a little wonky and lead to some variability. I think Ashwell had a question. You sure you didn't? I saw your hand move. Okay, that's fine. Yes, question over here.
Where do the A and the Z ions come from?
A and Z ions, okay.
Not the X ions
Yes. Okay, when a peptide bond breaks, B and Y ions are created. A B ion is kind of an unstable little beastie though. And when a B ion breaks apart, it may throw off carbon monoxide. And if it does that, it creates an A ion. Okay, now C and Z are a different case. C and Z are not peptide bond breaks. They happen one bond to the C-terminal side. And those occur when we do electron transfer dissociation or electron capture dissociation. But you should not see those in the case of HCD. Good question. We could have a whole hour-long seminar on how peptides break. I love the subject, but I know we may not want to do that in time. So we're going to talk about how quality control has started visiting other modalities of operating our instruments. We aren't always doing shotgunning. Sometimes we're doing some other places to go. So we're going to start by talking about the concept of the chromatogram. We're going to use some ancient figures from USP to teach us about them. Chromatograms figure to be very important in all kinds of quantitative mass spectrometry. So I think we have to talk about them. From there, we're going to talk about selected reaction monitoring, which is just a different name for the same thing as multiple reaction monitoring. MRM was a product name. It was a product name, sir. Anyway, people have religious feelings about these things. It's okay. So we'll continue with our discussion of SRM/MRM. And then we'll talk a little bit about SWATH. So if you'd like to debate it, we can. But maybe we'll save that for after second team.
It's really simple. Selective reaction monitoring is where you select one transition and you monitor it. Multiple reaction monitoring is where you select multiple transitions and monitor them.
A great many people disagree with you on that score.
It's not that. It's not. There's no debate about it. That's
Okay,
the reality.
I'm glad to know there's no debate about it. All right, so SRM is a valuable technology. We'll talk about getting quality control metrics from that. SWATH is a...
Can I just say, it's a bit like, that's a bit like the debate between is the earth round or flat?
I'm sure that no one has ever published a paper saying SRM can be used in multiple targets in a single experiment. Were they just wrong?
No, that's something different. Multiple targets is multiple precursors.
It's not multiple fragments.
It is multiple fragments.
SRM certainly is used for multiple fragments for multiple targets.
No, it's not. SRM is the reaction. One is one. That's the definition of SRM.
Okay.
Anyway,
I'm prepared to die on this.
Even Steve Carr, who was a proponent of SRM, agrees now that it's MRM.
Okay, that's fine. So we'll continue talking about selected reaction monitoring. We'll continue on to SWATH, a new way to use your triple TOF and other instruments that have high resolution. And we'll talk a little bit about MALDI-TOF profiling, because yes, those folks need quality control too. So let's talk about USP, our useful starting point. It's not a useful starting point. It's being used here as a useful starting point. But USP is the Pharmacopoeia of the United States of America, 38th revision. It's gone through all kinds of stuff. This is the document that defines a lot of how we characterize biomolecules in the United States. So they start with their definitions and interpretation of chromatograms. So the simplest way to think about a chromatogram is that it is a pattern of intensity as a function of time. Pattern of intensity as a function of time. So we see that we have our total experiment, we have maybe some solvent peaks that show up at the beginning of this, and then we have some feature that shows up that has this sort of quasi-Gaussian shape to it. And we see that we have various metrics that have been put together around it. Now, I would start by reminding you we were talking about chromatograms just a moment ago when we looked at the metrics from QuaMeter. You remember the full width at half maximum? So full width at half maximum shows up right here. We see that one of the ways that people compute width is to put a line down this side of the peak and a line down that side of the peak, and then compute the width as where those hit baseline. That is one way to do it. But a more popular approach asks what is the total height of the peak, and then we drop to half of that, and we say at this point at half the height of the peak, we ask what is our width. So when we talk about the full width at half maximum, we're asking how wide is this peak when we're looking at it at half its intensity, its best intensity. So this is one of the definitions that we can take forward from USP when we're working on this. Now there are a variety of different metrics that get computed within USP, like resolution. Is it okay to just make up your own definition of resolution when you're selling something? No, it's not. If you want USP to take you seriously, you had better be able to describe resolution under the definitions that they've put forward. Separation factor. Frequently we hear things like plates. how many plates of separation do we have in a column, for example. If you have more plates of separation, your ability to tell apart peptides that have very similar hydrophobicities is better. If you have very few plates of separation, you may fail to resolve them, and they may appear from the column at the same time. Most people would say proteomic LCM-SMS separations are pretty poor, frankly. The amount of the narrowness that we get of individual peaks is something like 30 seconds in a lot of cases. Now, the folks who are working with ultra-high pressure pumps, etc., can sometimes do it quite a lot better and quite a lot faster than what we can with HPLC. But generally speaking, we have an awful lot of ions, and they're not perfectly separated on this retention time plane. Symmetry factor helps us to ask about whether the peaks rise and fall at the same rate. More typically, we see sort of a lopsidedness to this, that the peptide ions rise very rapidly to their maximum and then tail off a little bit more slowly than they arrived. So we might describe this as a symmetry factor. The signal-to-noise ratio reflects how much a peak is able to stand out given the variations in intensity around it. And the percent RSD is another way that we can talk about variation in this context. So that's all I'm going to really talk about here, but these USP concepts have ended up percolating into our field as well, and they're worth knowing about. Now, if you're working in the space of SRM, and we'll just call it MRM for now as well, or working with extracted ion chromatograms as drawn from MS scans, you may want to have some pre-produced packages for making sense of these. Some of these have already been incorporated within tools like Skyline. Skyline is one of the tools that we'll use in this early tutorial. So SPROCOP is one from Mike Berriman, and it's a very valuable toolkit for evaluating SRM kind of data. And MS Stats from Mina Choi is also pretty great. Has anyone heard of Olga Vitek? An excellent biochemist, excellent biostatistician who has found her way into our fine world of analytical chemistry. So Mina was a graduate student of hers and put together this really amazing toolkit called MS Stats. So a lot of this has sought to integrate with Skyline because Skyline is so broadly used in making sense of this kind of data. There are a couple visualizations that show up very, very frequently within the space of quality control. And one of the first of these is called the Schuhart process control chart. This is something that I think that Marina might be able to define a little better for us. But the short version is that this is a univariate approach to evaluating the quality, the variability of a particular metric. So imagine that we simply look at the Berger counter. How many tandem mass spectra could you produce per second at the fastest rate within this experiment? So maybe you have 1,000 raw files that all represent BSAs that you've shot on your instrument over a space of many months. So for each of these, I can reduce each raw file to its Berger counter metric. What was the maximum number, the maximum frequency of tandem mass spectral production? So I can create this plot as a function of all this time. Sometimes it was a little higher, sometimes it was a little lower, but it's got some mean and it tends to cluster around that. Now imagine that some of these BSAs represent times when the instrument was really fouled up. And it might be that I see a variation that's way up, where I had a much higher tandem mass spectral collection rate than I would have expected. Maybe we double injected the standard, or maybe we injected a very tiny amount of it and the value dipped well below. So we see that we have a mean value that we can report across this collection of files. We have a +1 standard deviation or +2 standard deviation line that we can put above it, and we have a -1 standard deviation, -2 standard deviation line we can put below it. So I'm going to ask you a statistical question. This is where everyone can sort of look down if you like. It's all right, I'll still pick you. If you have a normally distributed metric, how frequently will you observe data that fall more than 2 standard deviations up or more than 2 standard deviations down? What fraction of the time should a metric that is normally distributed fall outside that range? 5%. You guys are doing great. Oh, no one had to look down that when you had that from a shot. That's great. Okay, so if you see a result that falls outside these control limits, we might think to ourselves, this could be a random event. So if 5% of your data fall above 2 standard deviations or below 2 standard deviations, probably no big deal. But it may be that you find data that show up outside 3 standard deviations. And something like that should only happen about 1% of the time. If that's happening 5% of the time, it's likely that a fair number of those are actual, there's something contributing to them being distributed differently than normal data. Okay, so the Schuhart process control chart represents one of the ways that we visualize a single metric over a lot of runs to give us some idea of what normal looks like. Okay, the next one of these that I want to show you is a Pareto chart. Pareto. So we have a number of things that could lead to odd experiments. We might have, it might be that retention time variation is the biggest contributor to files being abnormal. It could be that peak areas appear to be quite odd. It might be that the widths of these peaks, the full width path maximum, is the standout characteristic, or it might be that peak symmetry is the problem. You can see that these have been listed in an order from most frequent to least frequent. So in evaluating the failures within this experimental set, we ask what is the most common cause leading to that failure. Pareto charts let us get that information. So things like SPROCOP have brought this kind of standard visualization technology from quality control into our realm. Now, MS Stats has grown into a very complex piece of software. We see that although we might think of this as being something we use from SRM/MRM experiments, sometimes it's useful when you're trying to reconstruct your chromatograms from DDA experiments. After all, things like MaxQuant rely on our ability to compute an area for individual peptides that's reconstructed from the observations of it in MS1 scans. So you can see that the same package allows us to visualize the intensities across many DDA runs, or to do things like differentiation in the context of SRM-MRM experiments, or to look at things like SWATH experiments in order to pull those data out a different way. Now I'm going to draw your attention to a really cool PhD project going on right now, and this is something that spans South Africa. Stoyan Stoichev has been working with us from CSIR to try to create quality metrics in the context of SWATH experiments. So Marina has been working on this for a while, and we've already been working on quite a few different ways that we can characterize these metrics. One of the most straightforward things to ask is the method I'm using for SWATH evenly dividing the signal across the swaths? Right. So people have been arguing that you should have more windows in the middle of the M/Z range than you should at the ends, because there are more peptides at this sort of median M/Z value than there are at very low or very high values. So if you have a very wide swath at the side and a very narrow swath at the middle, that should be great. But it would be awfully nice if you could characterize how many ions appear in these ranges, or what TIC values appear in these ranges. So something like this could be useful for monitoring long-term use of swath. I've got one method, I'm going to stick with it, how am I doing? But they can also be very valuable for people who are getting started in swath, to be able to set up methods that best reflect the biochemistry of their samples. Okay, so things like that are quite valuable. Dot products. Dot products. I think you probably saw dot products during Stephen's talk yesterday. Dot products are frequently used as a way to evaluate whether a set of intensities most resembles what it should. So I would, an example that's going to show up in the earlier tutorial today concerns how intense the monoisotope is versus the isotope plus one neutron and the isotope that has 2 extra neutrons in it. So if you have a very small peptide, you would expect the monoisotope to be the big one. If you have a pretty large peptide, the monoisotope is no longer expected to be the biggest peak. So one of the ways to evaluate whether the intensities you see reflect what you expected is this dot product computation. Dot products are very popular also in spectral library analysis. In the case where you're trying to evaluate whether an observed tandem mass spectrum resembles the annotated tandem mass spectrum in your library. So dot products get used pretty widely. I'll So we think that there are a lot of opportunities then in data independent acquisition, precursor fragment correlation as well. This is maybe something that we don't think about a whole lot. If you live in the shotgun world, you're probably quite accustomed to thinking of, well, I have this tandem mass spectrum, and it was obviously seen in the preceding MS scan. So if I want to know what it's, which ion it corresponds to, I don't have to do any further thinking. I just look at the preceding MS scan. But with data independent acquisition, with SWATH kinds of experiments, we see that we have fragment ions that are associated with some collection of precursor ions. And that is a little harder to deal with. So in this case, the MS scans that we have in here along with our SWATH scans give us an appreciation of which precursor ions are available at a given time. And when we look at the tandem mass spectra, the SWATH scans themselves, these represent the fragments of peptides. One of the most frequent ways to associate a set of fragment ions with a particular precursor chromatogram is through correlation. The expectation that if this peptide ion is rising and falling in a certain fraction of time for the precursor scans, the fragments will rise and fall in the same time scale in the SWATH scans. So this association is what we do when, for example, we're using the software from SciEx to export an MGF that tries to make SWATH data look like tandem mass spectra collected by DDA methods. So there's a lot of room for evaluating whether this association is a strong one or a very weak one. If you have multiple precursors with barely differentiable chromatograms in the MS scans, and there's a set of fragments that come from these two different precursors, you might find it very difficult to discern which of the precursor masses corresponds to which fragment masses. Now, one of the things that I've been doing lately is working with a group on a strategy called MSQC, which is a very standardized way to pass quality control information around. So if you have a database tracking this information, we have standard ways to communicate it to downstream software packages. And along the way, we learned a little bit about this work from Lopez Fernandez in doing quality control for MALDI-TOF. Now, how many of you work in association with a MALDI-TOF platform? Okay, a couple folks back there too. All right. I used to be at Vanderbilt University, and they had a really strong MALDI profiling and imaging center there. So they were producing very, very large volumes of data. So some of the approaches that we see are as simple as taking a spot out of a 2D gel and trying to figure out which protein it represents, right? So you might spot this, take this protein spot, digest it, and let the peptide settle into a MALDI well, shoot a laser at it, see which peptide masses are there, and you might use those peptide masses to tell you the identity of the protein. Or you might identify individual peptides if you have something like a TOF. Okay, so Mass Up was designed to help people doing this kind of large-scale analysis. So things like the percentage of presence is a feature that they use quite frequently here. This is giving us the ability to say how reproducible is a particular peak across replicates of the same thing. It can handle a lot of statistical testing for you to be able to do differentiation for you. It can handle principal components analysis. There we see it showing up again. This is part of the the afternoon tutorial. It can do unsupervised learning through clustering. I think we've heard about that strategy a couple times already. It can do biclustering. Biclustering is a strategy that a lot of people associate with microarrays. In it, you try to figure out which samples have the most similar gene expression and which genes are most similar across patterns of samples. So in effect, you're grouping in 2 dimensions at the same time. So biclustering is feasible here. And even doing work with predictive classifiers. And predictive classifiers are hugely valuable in cases where you're trying to discern which mass spectrometry data are most useful to you in making a clinical decision. And I think you got a master class in that yesterday. So Mass Up tries to cover all of these capabilities, and happily, they're working with us in this MZQC effort. So I wanted to give you just a a quick reminder of how these things scale as we start thinking about reproducibility. So in mass-up, there are two fundamental types of mass-to-charge matching: intrasample and intersample. So intrasample, intersample, which of those applies to comparisons within a single replicate? Intra, very good. And so inter means we're comparing between different experiments. All right. The intrasample matching is applied to the spectra obtained for the replicates of the same sample, while the intersample matching is applied to match M/Z values across different samples, making them comparable and suitable for subsequent analysis stage. So this is a software suite that's really shooting for the fences, trying to give us a very broad set of capabilities in the context of MALDI-TOF analysis. And one of the things that they have to deal with is this problem of curbing missingness. Missingness refers to how frequently you're missing values for a particular peptide or particular protein. So you might imagine that if you were doing SRM or MRM on a very vanishingly small amount of some sample, it might be that some samples have nothing for you to integrate. You just get no observable chromatic data. For something like that, the software should have some sort of error code, presumably it can write, to say there's nothing that I can integrate here. It might, however, simply record, it would just integrate the noise, in which case you just get a very small value. So in the first case, you have a problem of missingness because your data table is not complete. And if you try to do a t-test, you can't feed it not applicable as one of the values, right? And so to use a very simple example. So percentage of presence is one of these things that the software tries to use, that MassUp is using to characterize the reproducibility of detection across many samples. Important. But what do we envision for the future of quality control and proteomics? I'm trying to tell this story in the context of some people, but I think that introducing the concept of decision support is very worthwhile. Quality control software provides the information required to select the best plan of operation for scarce instrumentation. That's my vision of decision support. And I don't remember what we do to make that door stop buzzing, but I'm sure somebody will be on that soon. All right, so we want technicians and lab managers and PIs to know what is the best way to use their instrument based on quality control metrics. So who does this help? We feel that this protects against wasted resources. And it's inappropriate to think of this as strictly in terms of money or samples, right? So some samples truly are irreplaceable. If you are not able to get this sample again, and you run it on an instrument that's operating in a faulty way, the data that are produced are not going to be reliable. That's a really bad scenario. If your column is blocked and you keep trying to shove stuff through it, well then that's not a great scenario either, right? So it's not always the expensive parts that are failing on you. Trainee time is precious. Trainee is kind of a terrible term, right? But it's generally the PIs are kept out of the room with the mass specs, right? You don't want us messing with these things, we're out of date and so on. The postdoc really knows what's going on. The grad student is very, very clever and sometimes devious, please so. The undergrad is probably not the person shoving samples into it, but all of these people are making use of this shared resource. And if you see that somebody's time invested in creating a sample and getting that sample run and analyzing faulty data from that sample, and all that comes to nothing, I mean, that's a terrible scenario. People have a limited amount of time, and seeing that time be on target matters. And certainly, damage to instruments is expensive. Now, I'm inclined to think here in terms of dollars, euros, and rand, but it's also in lost time for instruments. If you can detect that an instrument is having a bad day, it could prevent you from seeing the instrument having minor failures. And if you can recognize minor failures in time, you might be able to stop massive failures of the instrument. And ideally, maybe you can avoid that board blowing out and just being a terrible expense it's going to take months to fix. So we have time and we have money and we have people and we want all of these things to, and we have samples and we want all of these things to be preserved. That's why we have to care about QC. I've mentioned that QCML, or more recently the MZQC effort, part of HUPO-PSI, the Proteomics Standards Initiative, is really working in this space. And in a recent paper from Will Bittermy and friends, we put together a whole bunch of different roles that being able to pass around quality control information can play. The first of these is passing to storage. Who here knows what a LIMS is? L-I-M-S, LIMS? A fair number, that's good. Okay, a laboratory information management system. For a very large lab that's handling a very large number of samples, you're probably going to have some sort of database that keeps track of which sample corresponds to which person, and it's sitting in what freezer in which block. Now, if you're running a one-man show or a one-woman show, that may be a little harder to afford as an option. But frequently, we would like to be able to put some sort of seal of approval on data that pass muster. And if you can pass the quality profile into your database to say, these files were produced, they passed these quality checks, that's useful to you. We have depositing, repositories. What is the network of repositories that serves the proteomics field for mass spectrometry data? Oh, you're almost there, I can tell. Tip of your tongue. What is the network of repositories that we use in proteomics to store our raw files? Can someone name a repository?
GitHub.
Panorama. Okay, Panorama is good for storing SRM assays and SRM results. That's cool. That is one. But for shotgunners, what do we use? Pride. Okay, Pride is a European repository, and it is part of a bigger network called? Proteome Exchange. Very nice. Okay, so imagine a future where you're looking at a data set that's been published by some investigator, and they've got all these raw files up there, or all these WIF files, or all these directories, whatever they've shoved into the archive, and you want to know, well, how many spectra is this? Are all of these files equally useful to me? If you have some quality metrics that define what those data look like, you may be in a better place to be an intelligent consumer of the repositories. So we see this role in deposition of data, in characterizing deposited data as an important one. Certification. If you are to publish a data set, we have a long list of rules that we must follow, the MCP guidelines or guidelines used by any of the other journals, that we must satisfy in order to show that our identifications are in fact meaningful. It's a very long list. Feel free to look up the MCP checklist if you're curious about this. So there's a lot of rules that we need to follow in that. If we also need to characterize the raw data that we've got, it may be that having a standard way to communicate quality control could be a part of a certification that these data are in fact what I said that they were. Of course, analyzing and notification, the last two forks there, I realize those letters are too small, are quite important. We're going to be doing an example of this analysis in just a minute, and we'll move into the PCA-based one, a post hoc analysis, a bit later. But this notification one is one that I think is a really, really big deal. At some point, we feel that the information systems dealing with quality data need to be able to communicate with you, the person responsible for keeping this machine alive, if there are problems that arise. And that is a really tricky kettle of fish, so we'll get there in a minute. I'm showing a picture here of Kevin Shai, a friend of mine who runs the proteomics laboratory in the Mass Spectrometry Research Center at Vanderbilt University. So he has to oversee more than 30 mass spectrometers in one facility. This is not just a big clinical operation. It's not all a bunch of Q-traps. These are very different kinds of instruments. Q-TOFs over here, some GCMSs over here, triple quads, orbitraps. It's a nightmare trying to keep track of all the different capabilities and what the current status is on those. Imagine that at a glance, he could see a health report saying that this is the current mass accuracy achieved on our orbitraps and Q-TOFs. The signal-to-noise ratios in these defined mixes that we've run are currently sitting at these values for these instruments. Wouldn't that be handy? Now that is a bit of a pipe dream at the moment, but I'd love to think 10 years from now, such systems would be in widespread use for these major mass spectrometry facilities. It would also be very nice if we were able to capture something that's being soft captured now. When a technician runs a calibration on the instrument, it's probably written down in a lab notebook somewhere. But how easy is it for me as an informaticist to look through that lab notebook and plot a time course of when these events took place? Do I know the date of the last time this instrument had a major service? Do I know when it was last baked out? Maybe I don't. Do I know when the software changed? Maybe the software version changed by 0.1 and suddenly experiments that were working great looked like trash. That would be alarming. Right now, we do not do a very good job of capturing this metadata about how the instruments are maintained. If we can do that, I think that we will find that people like Kevin Schei's lives become much better. They can have a roll-up list with a little green light on all the instruments except red on that one instrument that's really just been a lemon since we bought it. Now, there was an article not that long ago that characterized people who use other people's data to write new papers as data parasites. Data parasites. Do we have any data parasites in the room?
One knows.
Bioinformaticists,
Yeah.
biostatisticians, yes.
Yeah.
Unite! You have nothing to lose but your fears. No, I know. So, in this case, I'm using a picture of my friend Johannes Griss as an exemplar of the data parasite. It's very rude. He's a really nice guy. It's so smart. But I want to point out that sometimes Johannes wants to dig through all of Pride. So he spends a lot of time looking at Pride. He wants to look through all of the Pride repository, and he wants to identify instances of common chemical contaminants that have tandem mass spectra and retention times and accurate masses that have been seen time and time again, and yet they just look like unidentified tandem mass spectra to most experiments. Wouldn't that be powerful? To go sniffing into the dark matter and come away with IDs that really do show up all over the place. So if he could do something like that, that would be powerful. But let's say that he wants to grab out all the data sets produced on Fourier transform capable instruments that had a large number of +1s. Now that's kind of contrary, because generally speaking, when we operate our Orbitraps and Q-exactives, we're telling them we don't want to look at anything +1, and so we don't capture those spectra. So this is kind of an odd bird he's looking for. Data sets from high-resolution instruments that allowed the acquisition of +1s. If the repositories had this huge set of quality metrics that had these kind of counts available to us, that would be very powerful. You could say, the fraction of spectra in this collection that were +1s was 15%. All these others are zeros, but this one's 15%. This is one I want to include in my paper. So being more intelligent consumers of the repositories would become possible through the use of quality metrics. Now, some of us operate in the trenches. Some of us have to work for a living. And some of them must struggle with instruments mightily to make them behave themselves. So here, I'm showing my friend Khadija. She must handle samples and instruments for a university core facility. This is not an easy place to live, is it? Her clients want their data when? Yesterday. Yesterday. So that's not convenient. PIs who have a grant proposal deadline tomorrow, they don't really find much interest in saying that the instrument is feeling sad today. You know, they want their data now. So that's inconvenient. Her inbox contains an email from the lab LIMS informing her that sensitivity has dropped on the ion trap. It proposes that she tries to claim the source. Okay, that's maybe nonsense, but imagine if you had a decision support system powerful enough to recognize that not only are these quality metrics aberrant, but the likely solution is this. That's powerful. So we need systems that can automatically generate metrics on the fly from the data that are coming off of our instruments. They need to have some sort of instrument history profile to compare these new data points against. And it needs to be able to say, we've now reached a critical point that needs to be addressed through remedial action. Okay? Now, what happens if the decision support system sends her email 5 times a day, whether the instrument is happy or not? Khadija, are you going to pay attention to those emails? No. Computer-generated emails do not get read at high frequency. So we better be sure before we pull the trigger on this decision support system that if it's sending an email, it actually has some basis for doing so. Okay, now, not everyone I'm interested in talking to about this works in proteomics at all. Oliver Fine has been working in metabolomics for quite some time. And imagine that he is writing a review article now on all the different platforms that are used in metabolomics. Is it possible at present to compare the coefficient of variation for lipid quantitation between QTOFs and Orbitraps using this huge backlog of data that's been shoved into repositories over time? At present, that would be very, very hard to do. So we need to have things like platform-independent QC metrics. If I write metrics that are only useful in the context of thermo instruments, that's not going to be of any service to people who are running triple TOFs. So having this capability matters. Being able to generate quality metrics that are agnostic as to what kind of sample is being run is also valuable. And the ID-free metrics that we're talking about today will be an example of that. And from this, we're also going to need a bunch of buy-in. Getting repositories to add new kind of analyses to their data is not an easy push. These are many terabytes of data to dig through in order to produce these metrics. So we need to make sure that we have some sort of payoff for them. There are lots of challenges along the way. There's the human interaction problem. If somebody has to call a service person, that's a big ask. If someone needs to change a column, well, maybe they have one on the stores, maybe they don't. Right now, these kinds of interaction are not really captured in a machine-readable way, and we need that. The actors, unfortunately, are very busy, and asking them to just sit down with their lab notebooks and make a jot in this database for every time that the column was changed over the last 5 years is a bit of an ask. It's a tough thing. Instrument data are stored in many places and are named inconsistently. If you are trying to create an information system that is triggered every time a QC sample has been shot on the instrument, it needs to ignore experimental samples and wait for the defined mixture to run again. So if people don't use any sort of naming system that makes it clear which ones are QC samples and which ones are not, we're doomed. So that's important. The other thing is that a lot of instrument facilities don't have proper file server support. One of the facilities that I've had to work with here in South Africa had its data, its backlogs of data stored on a bunch of external hard drives. And to get their archives put together, I had to track down where all of these hard drives had migrated over campus. That is stressful. So the IT services that support a lot of laboratories are not currently ready to help us in this QC task. And seeing that those infrastructures grow matters. And we need systems to learn typical behavior of MS and to diagnose variances. We'll talk about some of those coming up. Our software will be deactivated as soon as the first person in the lab feels pestered. If we're going to roll out decision support, we have to be sure that its messages are worth listening to. And this is one of the things that shows up again and again when you look at software implementations, say in healthcare. If you start pestering people with emails, your software is toast. So we have a lot of challenges to face to get to our dream. So essential work, like QC, seems a little bit less glamorous. I would love to be talking to you about arbitrary post-translational modification identification today. I would love to be talking to you about ways that we can increase identification speed by an order of magnitude. Those are kind of the things that bioinformaticists just love doing. But in fact, QC is an area that's awfully essential. And even if some people find it a little dull, it's actually extraordinarily useful. So I would note that some of the things that we think about for our research career may be whether these things interest us, but in fact, if they just have to be done, sometimes that's all the justification needed for pursuing that as a line of research. Proteomic paradigms beyond data-dependent acquisition and selected reaction monitoring need more support in QC toolsets. This is coming along, not as quickly as we would like and not as popularly as we would like, but I think that we will get better in this space. And certainly quantifying variability is many, many steps away from actionable advice. If I can provide a technician information that lets her decide how to best keep this instrument running, that's valuable. But if my software starts being noisy and ignored, I've lost my opportunity for that. So we have to do it right on the first time. Okay, so those were my closing thoughts. That's my dream of where we can go with quality control. But in the next sections, we're going to get a more realistic portrayal of where things sit today. And I hope that it will help you to understand what we can accomplish now and where we need to improve.